All right, let's get right into it. We're about to go inside one of the most exclusive and, frankly, most brutal training programs in the entire universe. And no, I'm not talking about the Navy SEALs. I'm talking about the elite academy that trains your body's most critical soldiers, your T cells.
Think about this. Right now, as you're listening, your body is recruiting, training, and arming a microscopic army. These are the cells that literally stand between you and a world full of deadly pathogens. But the training to get there, it's unbelievably harsh. In fact, the failure rate for recruits in this training program is a mind-boggling 98%. That's right, almost every single recruit is eliminated before they ever even see a battlefield. So what is this insane cellular boot camp? And why on earth is it so deadly? Well, let's find out. Okay, first things first. Our story has to begin with recruitment. We need to figure out who these cadets are and where this whole intense training program actually takes place. So the recruits themselves are called thymocytes. You can just think of them as young, uncommitted cadets that show up from the bone marrow. See, they're not really T cells yet. They're more like blank slates. They've got the potential to become these elite soldiers, but man, they have a really long road ahead of them. And this right here, this is the academy, the thymus. It's a small organ right in your chest. What's so cool about this slide is how it maps out the whole journey for a recruit. They show up, they move into these outer training fields. That's the cortex. And this is where most of that brutal selection goes down. If, and it's a big if, they survive, they get to move to the inner headquarters, the medulla, for their final exams before they can graduate. So once our cadets are officially in the academy, it is time for basic training. This is where they fully commit to becoming a T cell, and probably most importantly, they have to build their primary weapon. This first phase is broken down into 4 stages, and they're called double negative stages. That's just because at this point, the cells don't have their key markers, CD4 and CD8, which you can think of as the insignia that'll later show what their specialty is. So they arrive, they commit to being a T cell in DN2, and then boom, they hit their first massive test in DN3. It's a quality check called beta selection. If they pass, they get to multiply, creating a whole platoon of cadets that all have the same basic gear. And this visual illustrates that first test perfectly. You see, a T cell's main weapon is its receptor, the TCR. It's what it uses to identify enemies. At this stage, the cadet has to prove it can build just one half of that weapon, the beta chain. It then pairs it up with a sort of stand-in piece to form what's called a pre-TCR. If this thing sends the right signals, it passes beta selection, gets the green light, and moves on. It's the first huge hurdle they have to clear. Okay, we've now arrived at the very heart of this training program, what I'm calling the crucible. This is where that shocking 98% failure rate comes from. Every single cadet who made it through basic training now has to face 2 final exams that will decide if they live or die. So here's the crucial part. There are 2 big tests, and they seem to completely contradict each other. The first is positive selection. The drill instructors show the cadet the uniform that their own body's cells wear. These are called MHC molecules. The cadet absolutely must be able to recognize this uniform. If it can't, it's totally useless. But then comes negative selection. This test checks if the cadet reacts way too strongly to that same uniform when it's carrying little pieces of your own body. In other words, are they going to attack their own troops just for wearing the right uniform? This slide shows you the brutal reality. The cadet meets one of these instructor cells. If its weapon, its receptor, can't recognize that friendly uniform at all, well, it's useless. It doesn't get a survival signal, and it dies by neglect. Gone. Now, if it recognizes that uniform and the self bits on it too strongly, it's considered dangerous. It's ordered to self-destruct. That's negative selection. It's only that tiny, tiny fraction that recognizes the uniform with a just right, sort of weak handshake that gets the signal to survive. That's positive selection. And look, this is not me being dramatic. The source material is very clear. About 98% of these cells that have worked so hard to get this far, they fail these tests and are simply eliminated. The thymus is basically a graveyard for the overwhelming majority of its own recruits. But wait a minute. This brings up a huge question, right? A paradox. You have to recognize self to pass the first test, but then you're killed for recognizing self too well in the second one. How on earth does any cell pass both? How does this academy not just kill every single one of its recruits? And the answer? Well, it's actually beautiful in its simplicity. It's all about the strength of the signal. It's about affinity. It's not if you bind, it's how strongly you bind. As this chart shows, it's a total Goldilocks situation. A grip that's too weak, death by neglect. A grip that's too strong, you get deleted. Only that just right, intermediate binding strength, strong enough to prove you're useful but not so strong that you're dangerous, allows the cell to be positively selected and to survive. It's an absolutely incredible balancing act.
Okay, so for the very few cadets who actually survive that great selection, there's one more big step before graduation. They need to be assigned a specialty. Are they going to be a battlefield commander or a frontline assassin? So how's that decision made? Well, it's pretty elegantly explained by what's called the kinetic signaling model. It all comes down to the duration of the signal they got during that test. If the cadet's receptor bound to an MHC class II molecule, it got a continuous, steady signal. That instructs it to become a CD4 helper T cell. Think of it as a field commander, coordinating the whole immune response. But if it's receptor bound to MHC class I, the signal got briefly interrupted. That little interruption is the cue to become a CD8 killer T cell, the assassins that go out and hunt down infected cells one by one.
We are at the final stage. The few surviving cadets have passed the crucible, they've got their assignments. Now it's time to graduate, get deployed, and make sure there are systems in place to keep this powerful new army from getting out of control.
Now here's a really cool twist. The Academy actually has a failsafe. It turns out that a small number of those cadets that had a high affinity for self, the ones that were supposed to be deleted for being too dangerous, are instead recruited into a special forces unit. They're called regulatory T cells, or T-Rex, and their job isn't to fight the enemy. Their job is to be the military police, to actively keep the other T cells from getting too aggressive and causing friendly fire. And these peacekeepers, they have a whole toolkit for maintaining order. This slide shows how they can basically starve out overactive T cells by hogging all the essential resources they need to grow. They can release inhibitory chemical signals to tell them to calm down. And in extreme cases, they can even kill a fellow T cell that's gone rogue. These guys are absolutely critical for preventing autoimmune diseases. So, after all of this, what's the big takeaway? Why does any of this matter? Well, because this entire incredibly picky, brutal process is the reason you're alive and healthy. It's the system that allows your body to launch a devastating attack on something like a flu virus while leaving your own healthy lung cells completely untouched. It is the biological genius behind telling friend from foe. This training academy is, well, it's nearly perfect, but it's a biological system, right? And nothing in biology is ever 100%. So it leaves us with one final and pretty chilling question. What are the consequences when this system makes a mistake? What happens when one of those dangerous self-reactive soldiers manages to graduate and slip through the cracks?